Okay, good morning, everyone. So congratulations on getting through our exam last week. Um, I believe we should have the grades for that released very soon, if not already. Um, if you have any concerns about your performance on the exam, I'm always here and happy to talk with you about where you're at in the course. It's still early in the semester, so there's plenty of opportunity to kind of think about different strategies for studying and keeping up with the material if you find that you're not at where you would like to be, right? So reach out. I'm happy to meet with you one-on-one to talk about where you're at. Okay, so let's go ahead and move on with our discussion. We had introduced the topic of tumor suppressor genes and tumor suppressor proteins when we were learning about oncogenic pathways such as the PI3 kinase AKT pathway. Remember, and we had learned that that pathway is dependent upon the formation of PI3 or PIP3 on the underside of the cell membranes to be able to attract AKT, resulting in AKT's phosphorylation and ultimately the activation of many pro-survival, pro-cell cycle types of phenotypic changes in the cell. We described that there was a protein called PTEN, a PTEN that reverses the process of PIP3 formation, turning it back to PIP2. And we looked at a slide that I think illustrated very nicely how defects in PTEN could result in things like out of control AKT signaling. Right? So that was our first example of something that would counteract an oncogenic pathway. And so that was our first example of something we might call a tumor suppressor gene or a tumor suppressor protein. So let's explore that a little further now. And we'll take a look at the recessive nature of the cancer phenotype and look at another very important example of a tumor suppressor gene, the RB gene. Right? So let's get back on the same page here. Just as a quick review, we said that upregulation of mitogenic signaling pathways are what drive proliferation of the cell. And these mitogenic signals might be coming from the outside in the form of different types of signals like epidermal growth factors or other types of signals that the cell interprets as messages to survive, to start moving through the cell cycle and start proliferating. Right? We learned that mutations or overexpression of certain proto-oncogenes can activate oncogenic activity in a dominant fashion. Right? In other words, if you have certain mutations or way too much of a particular receptor tyrosine kinase on the surface, although that itself is a result of a mutation somewhere, right? A mutation that doesn't necessarily control the coding sequence of that RTK, but the expression level of that RTK. When those things happen, right, it seems like you get a very dominant oncogenic type of change happening. Right? So what we are interested in talking about now is, even though it's exciting to talk about oncogenes, because that's what we've been focusing on for the first few weeks of this class, just as exciting and I think thought-provoking are what are the mechanisms that are in place to normally keep those potentially oncogenic pathways in check, right? Or under control. So how do these tumor suppressor genes normally prevent cancer in healthy cells? So this is a very another example of a very early classic experiment where oncologists in the lab, decades ago, were trying to figure out, these changes that happen that we associate with a cell transforming and becoming an aggressive cancer cell, right? How do these really kind of relate to the mechanisms that are in that cell that normally should be in place to keep these changes from happening? So one experiment that they did was they would take a normal cell of some type, right, and they would have a cancerous version of that same cell type. And so really the only difference here is maybe you have, you're growing these cells in a culture, right? Some of them you transformed and they've become now kind of crazy cancer cells, but you still have the normal original cell that they came from. What happens if you take one of each and you fuse them together? This is kind of neat, right? You fuse them together and you imagine the resulting cell would probably do one of two things. If the hybrid cell is still tumorigenic, in other words, it's still kind of crazy and proliferating out of control, you might go ahead and conclude, well, the cancer alleles, whatever is changed here, changes in the genes due to mutations, those are dominant, right? Or if you take those hybrid cells and you look at them and you realize, oh, the hybrid cells have begun to lose their aggressive cancer phenotype, right? They've stopped proliferating. Then you would conclude that the cancer phenotype or the cancer alleles are somehow recessive, right? That once you've sort of restored some other component in here, it doesn't matter that you have the cancerous alleles, the cell stops becoming cancer. So which one do you think they saw? As it turns out, what they saw was the latter, right? That when they would fuse cells together, more often than not, the cells would lose their cancer phenotype, right? So when tumor cells are fused with normal cells, they lose the ability to form tumors. Therefore, the conclusion is that the malignant cell phenotype is recessive to the phenotype of normal wild-type cells, right? So in other words, this was the first evidence that there must be things that are present in the normal cell that keep things in check, keep these oncogenic pathways turned off, and that these, whatever these are, have somehow gotten lost during the transformation into cancer, right? So that if you then fuse them back together again, you're essentially restoring the presence of these tumor suppressor factors in there. So the one exception that they would see in these types of experiments is when the tumor cells arose from some sort of oncogenic tumor virus infection. For example, the addition of VSARC through Rous sarcoma virus. In those types of cases, then the cancer phenotype still dominated. And I ask you to think about this. How would you explain that? And hopefully what you realize is because when you introduce something like a mutant form of an oncogene that has mutated so that it no longer responds properly to the tumor suppressor factors that are in the cell to keep it shut off, right? Something like the Rous sarcoma virus or the VSARC, right, can then really still function independently compared to the different types of repressive mechanisms that a normal cell would have inside of it. So when early researchers realized that there must be certain genes that are involved in coding for proteins that are tumor suppressor proteins, right, they began to wonder, what's more important, right? The proto-oncogenes mutating into full-blown oncogenes, right, or tumor suppressor genes being somehow disabled? Because there was a certain group that from the start argued that these tumor suppressor genes must be an important factor in the progression of cancer, because it must be that the loss of these tumor suppressor genes are a critical part of cancer forming. Why? Because if you think about the nature of random mutations, if you have a coding sequence of a gene, any gene, right, that codes for a functional protein, and you have random mutations that you introduce in the coding sequence that might cause missense or nonsense substitutions of the amino acid identities, it's way easier to imagine that random mutations would hinder the normal function of something like a tumor suppressor gene rather than random mutations magically enhancing an oncogene's function. Right? Therefore, this group of researchers argued that it makes more sense that mutations lead to tumor suppressor genes being switched off rather than oncogenes necessarily being turned on, right, as the basis of how cancer happens. Right? their argument is this. If you think about this, and I think this actually makes sense. Let's say you have a key, and a key represents a three-dimensional shape that's very specific, right? I mean, that's the nature of a key. The little grooves, the little teeth, things in there are very specific to work in a particular lock, right? If you were to randomly damage your key, which is sort of like randomly causing an amino acid substitution in a protein that alters its three-dimensional shape even slightly, ask yourself, is it more likely that this damage would result in this key failing to work, right, on its own lock? Or is it more likely that a random mutation, random damage like this would somehow give this key the ability to have, like, additional properties to open other locks? Right? Hopefully you'd realize, yeah, random damage to a key is more likely to cause it to fail to work rather than giving it new properties or new powers, right? So that was the argument here, that maybe the development of cancer has more to do with the loss of tumor suppressor genes than it does the introduction of mutations that cause oncogenes to function. But there was another camp of researchers early on who argued against this, right? Because they said, no, wait a minute, think about it, right? Random mutations, if we're talking random, it's very unlikely that you'd end up getting two random mutations, each hitting the same tumor suppressor gene, right? Because remember, we have two copies of things like our tumor suppressor genes, one copy that came from mom and one copy that came from dad, right? So even if you had a random mutation that messed up the tumor suppressor gene that you got from dad, right, you'd still have the one from mom. So what are the chances that another random mutation would disable that one? That seems very unlikely, right? To get two hits that would actually disrupt and eliminate the function of a particular tumor suppressor gene from one of your cells. So if tumor suppressor genes act in a genetically dominant fashion, then both copies of this gene must get hit with disruptive mutations to allow cancer to progress, right? That was the argument that was being made there. And the likelihood of this seems very unlikely, right? So you have a random mutation that hits this particular chromosome right here, and this is where, for argument's sake, a particular tumor suppressor gene is, right? But you still have another good copy of it. Really, what are the chances that you have another random mutation that ends up damaging the other copy, right? We can think about it this way. The likelihood of any individual base pair of DNA throughout your entire genome. Remember, our entire genome is made of 3.2 billion base pairs of DNA. Right? If you were to randomly close your eyes and put your finger down and pick one base pair of DNA somewhere on some chromosome, what are the odds of a random mutation happening right at that spot? Right? It's astronomically small. Right? So they've calculated it. The risk is about 1.1 times 10 to the minus 8 chance of a hit randomly happening per cell generation, right? Meaning that every time the cell divides, right, you can kind of revisit this and say, okay, now what are the odds? Well, it's still 1.1 times 10 to the minus 8. That's really small. An order to odds of this happening, not only to that one base that you've singled out, but to then have it happen again, right, within the gene on the other copy, on the other chromosome. So that was the argument against tumor suppressor genes having to be that important, right? Because it seems very unlikely this would happen. However, as time went on and researchers began to examine different types of cancer, they realized more and more that this is indeed what is happening. So a really good example of this comes from the study of this particular type of cancer known as retinoblastoma. So retinoblastoma is a solid tissue mass that forms from the cells of the retina, right? So you end up with a solid tumor mass that ends up causing blindness, and if unchecked and undetected, can spread. So here is a advanced case of retinoblastoma where the eye is essentially almost fully just this mass of cells. And importantly, these cells have now started becoming malignant, and they're migrating into the optic nerve, right? So the tumor is spreading beyond its normal primary location. So retinoblastoma affects about 1 in 20,000 children from between the ages of birth to 8 years old. So it's a particularly sad type of cancer because this is a cancer that affects kids, right? And really predominantly kids. So there are two forms of retinoblastoma. There's what we call the familial form and the sporadic form. Sporadic, as the name implies, sporadic sort of seems like out of nowhere, right? Sporadic. This is when the child presents in the clinic with one eye having a tumor in it. And upon removing or treating that tumor, there is no elevated risk of the other eye developing a tumor, nor is there any elevated risk of that child developing tumors elsewhere in the body, right? So that's the sporadic form of retinoblastoma. The familiar form is when the child presents in the clinic with tumors in both eyes. Right? And unfortunately, in this case, in this type of the disease, when you remove the tumors or treat them, it doesn't offer any protection, right, against tumors forming elsewhere in the body. And in fact, children who have familial retinoblastoma, they get tumors elsewhere. Right? So that's a big distinction, right? So what's going on there? Right? So let's look at this data. Here is some very telling evidence that something very distinctive is going on here between these two forms of retinoblastoma. They took a cohort of kids who were diagnosed with retinoblastoma, and they broke them into two groups. Those kids who presented with unilateral tumors, in other words, only in one eye at the time they were diagnosed, versus kids who, when they were diagnosed, there was evidence of tumors in both eyes, bilateral. Right? And so these kids were treated, right? And so this is a Kaplan-Meier plot following these two groups of kids, retinoblastoma patients, in years after they were first diagnosed with retinoblastoma. And on the y-axis, the clinical endpoint that's being measured here is whether tumors ended up appearing somewhere else in that person's body as they grew out of childhood into adulthood. Right? And what you can see is very striking. Right? Those kids who presented with retinoblastoma, the unilateral form of it, where it was only in one eye, if you follow them decades later in their life, right, only about 5% of them ended up having tumors anywhere else in their body. And that percentage is no different than any random person from the general population. Okay. Yet, if you follow those kids who presented with retinoblastoma in both eyes, look, decades later, over a third of them had tumors elsewhere in their body and the formation of cancer somewhere else. Okay. So that is really striking. Okay. So what is going on there? So let's look at this, the dynamics of retinoblastoma formation. In the case of familial retinoblastoma, familial means that there's evidence, right, that this is passed on through the genes within that family line. You can imagine that the child is born with one defective copy of the retinoblastoma gene, right, or the RB allele is a mutant one on the particular chromosome that codes for RB, right? Let's say that he or she got this copy from dad, right? And then he or she got this copy, the good wild type one, from mom, right? Every single cell in her body will have one copy of the mutant RB allele, right? Because it was inherited from the dad's side, the dad's contribution to her genome. So every single cell in her body has this mutant RB allele in it. But when she's first born, every single cell in her body has a good wild type copy. Right? And so that wild type copy can produce the good RB protein, the wild type protein. However, that means that all it will take is one hit in the good copy of the wild type RB allele to completely then knock out the function of the RB protein in that particular cell that gets that hit. So in this case, it is almost inevitable that somewhere along the line, right, in the first few years of life, there will be a cell in the eye that is already carrying the one hit with it as part of what it inherited. It is almost inevitable that one cell somewhere in the eye will get a second hit, right? And that second hit will knock out the RB function, and then that cell will begin to transform. Right? And that's why in familial retinoblastoma, it happens in both eyes, right? Because both eyes, every single cell in them already have one hit to them, right? So it is almost inevitable that if you're talking about billions of cells in the eye, right, that somewhere in there, a cell from each of the eyes is going to get a hit in the one remaining good copy. Right? And this is the thing that's amazing. This is why it happens early in childhood, because you think about it. If every cell in your body has the version of the RB that you inherited from mom and from dad, why is it that the tumors develop in the eyes then in RB patients? It's because the cells in the eye, by design, are exposed to much higher levels of UV radiation, right? Our eyes are designed to, on purpose, let in tons of sunlight, right? So the cells of our retina have very little protection against UV radiation. So even though a child who's born carrying one mutant RB allele has that mutant allele everywhere else in their body as well, it's in the eyes, right, that they will first encounter the elevated mutation rates that will cause cells in each of the eyes to develop the disease. And that's why it's bilateral. And that's also why that by developing familial retinoblastoma, it is a sign that later in life, all of the other cells in your body are also carrying, this one hit already, so that there's a much higher increase in probability that somewhere else in your body through decades of life, you're going to develop another hit that results in tumors. Okay. Compared to sporadic retinoblastoma, right? So in sporadic retinoblastoma, when there's no family history of carrying a mutant RB allele, right? You're born with two good copies of the RB. Hey, your eyes as a kid were just as exposed to the UV radiation as the poor kid who developed RB, right? So how come you didn't develop it? Well, it's because, again, random mutations mean if you picked a particular cell in the retina of your eye, right? Let's say that particular cell got a hit in one of its RB alleles, right? You still have another good one, right? The other copy that you have is good. So both of the eyes are still good, right? And then on the minuscule chance that that cell actually then gets a second hit to knock this out, right, to actually cause that cell to start forming tumors, right, that's only going to happen probability-wise in one eye. And that means if you were to treat that tumor and get rid of it, right, you're in the clear because all other cells in your body, right, have the normal wild type retinoblastoma, right? So that's the big distinguishing feature here. I know that sounds a little complicated, but go back, talk your way through it, and realize this was a critical piece of evidence historically linking tumor suppressor genes and the loss of tumor suppressor gene functions to the development of cancer. So I'll tell you a story. So one of my close family friends who I grew up with in Boston, she's also Chinese American. And at the time in the early 80s in Boston, there weren't many Chinese American families, right? So we all tended to kind of or at least our parents tended to be good friends and stuff like that. And then, us kids, we sort of had to be friends with each other because we're dragged around by our parents to these different types of get-togethers and things like that. So we've kept in touch over the years. So several years ago, she was visiting Hong Kong with her daughters, and her husband, because her mom is back in Hong Kong now. And over the Christmas break, they were hanging out together. So her sister was also there, and her sister is a physician. And when her sister was playing with, her daughter, or my friend's young daughter, who was only 4 years ago, her sister, who's the physician, looked at her eyes and realized she saw something in one of them, right? That really, really alarmed her. And so they quickly took her to a hospital in Hong Kong where she was diagnosed with retinoblastoma, right? So they saw a tumor forming in one of her eyes. Okay, so this is no family history of retinoblastoma at all. It was happening in just one eye. So this is classic sporadic retinoblastoma. And so it ended up, my friend, she had to make a very difficult decision with her husband. Their daughter was like 4 years old. And so it was either have her daughter undergo really intensive chemotherapy to try to spare the eye and keep the eye, but target the tumor and get rid of it. But there would always be the risk, right, of the tumor coming back if they didn't fully get rid of it. And there was always the risk of these intensive radiation therapy, chemotherapy, of causing lasting damage, right? Because after all, the girl is only 4 years old, right? She has potentially decades of life ahead of her, but the only other alternative was to physically remove her eye, right? So, oh my God, what a terrible decision as a parent to make, right? Concerning a 4-year-old. So ultimately they chose to remove her eye. And so, so as a 4-year-old, she was fitted with a prosthetic eye. What is amazing is that these current modern prosthetic eyes are amazing. You cannot tell that it's a fake eye. But as she's getting older, right, I think there will be 2 or 3 times in her life where she actually has to replace and be refitted for a new prosthetic eye. But the amazing thing about this, and the really sad thing about that, is they realized when they looked through the holiday photos and family photos of the previous couple years before that happened, that in many of the photos that had their girl in it, they could actually see a little glint in one of her eyes in the photos, right? And they never thought about that. But now they realized that that was actually the tumor reflecting the flash from the camera, right? So anyhow, I know that's a real downer of a story, but fortunately, that is sporadic case. So now that she's had that tumor removed, right, there is no worry and no fear of increased risk of having RB-related tumors elsewhere in her body as she grows and becomes an adult. Okay, so where is this RB gene located? So RB is located on chromosome 13 on the Q arm, so 13q14. Here is an image of one of the very first karyotype images that was crucial in understanding what was causing retinoblastoma. And in this karyotype image, here is the pair of chromosome 13s in the 6-year-old retinoblastoma patient, right? This is from decades ago. That's why back then, this is the best they could do, right? These grainy kind of images. But you can tell when you compare the 2 copies, the one that came from mom, the one that came from dad, or whatever, I don't know which is which, right? You can tell there's something funny going on, right? Because they should look identical. Right? Here is chromosome 13, right? So chromosome 13 should have this banding pattern through this tiny little minuscule P arm, which is really nothing. Most of chromosome 13 is actually the Q arm, and it would have this distinct banding pattern shown in green here, which is the pattern you see up here on the left. But notice the chromosome 13 on the right is different, right? Something has happened. And what they realized is that there is a region of chromosome 13 that is missing, right? And that's why you don't have this extra green band at the top here, right? There's this big empty space and you don't see the band there, right? So it's a deletion. It is a loss of this region of chromosome 13, and they realize that the gene that is somehow responsible for the formation of retinoblastoma must be encoded for in this missing region, and that's why they named the gene RB. Right? Okay, so in this particular case here, though, if this came from a 6-year-old RB patient, what does that tell you about the status of the RB gene in the other chromosome? You say, well, it's not missing, right? Because there's no deletion there. It looks like it's still the normal pattern. But wait a minute. this is an RB patient, right? That means this other copy of the RB is the second hit, right? This little 6-year-old boy, I think, was born with a deletion of chromosome 13. And within the first few years of his life, right, it was inevitable that in some cell in his eye, there'd be a random mutation in the other copy over here that gave rise then to his disease. So, is this other copy then wild type in this boy? No, right? This other copy must have a point mutation also. Otherwise, he would not have developed the RB, right? And I should mention, this is, of course, taken from the actual tumor cells. All right, so what does RB do? So the protein product of RB, or the RB protein, sometimes you'd see it just called PRB, it prevents cell cycle progression. Right? So here you have an illustration that is showing you that there are certain genes in the genome, this target gene here, that is some gene that's involved in encouraging the cell to move through the cell cycle, right? So normally a fully differentiated cell that's happily just doing its normal fully differentiated cell function, for example, a retina cell, right, that's in the lining of your retina, maybe the rods, the cones, whatever, it's just doing its normal job as part of the retina, right? It's not concerned with proliferating, etc. So these genes are normally shut down, right? They're not active. These genes are normally activated by the presence of a family of transcription factors known as E2Fs. So if these E2F transcription factors are allowed to bind to their target genes, then they will activate these target genes and the cell will then start moving through the cell cycle. Right? So these E2Fs, though, are usually sequestered or kept turned off by being bound to the RB protein. So you can see here, the PRB, the protein product of the RB gene, right? It is a protein that sequesters or hangs onto the E2F transcription factors, keeping them out of the nucleus, keeping them from turning on those cell proliferation genes. Okay? So what normally happens then is if the cell does receive signals from other cells telling it, you know what, you should go ahead, take a break from doing your normal differentiated functions and start doing, moving out of G1, start going into S phase so that you can start doing mitosis. Let's look at the left here. That would be if you have different growth factors or different mitogenic factors arriving on the outside of the cell, right? These growth factors, as we learned, would be transduced by receptor tyrosine kinases, right? And these, like the EGFs, for example, or the EGFRs, right? Because they're the receptors. And then that will result in the expression or the activation of certain types of genes that are now going to control other downstream functions to start driving the cell through the cell cycle. So one of those is the production of different cyclin and cyclin-dependent kinase proteins. These cyclin and CDKs we will learn about a bit later when we talk more about the cell cycle specifically, but what they do is they target the retinoblastoma protein and they phosphorylate it. Once the RB is phosphorylated, that forces the RB to change its shape, right? Because remember, phosphorylation is one of the most common ways of post-translationally modifying a protein to sort of turn it on or put it in an active conformation or sometimes put it in a turned off conformation. In this case, right, RB lets go of the E2Fs, and then the E2Fs can go and start turning on those genes that are part of making the cell cycle move forward. So if you don't have RB, or you have a defective form of RB that's not the right shape to start with, these E2Fs are allowed to just start working without this important control mechanism, right, keeping them in check. So that's why RB is a very important regulator of the cell cycle. When we talk about this idea of having two copies of RB, one from mom and one from dad, right? And meaning you would have to have two hits or two sets of mutations that both hit one of the copies of RB in order to knock it out, right? Because if you only have one hit that disables one RB, either deleting it or making a point mutation that messes up the RB protein that comes from that copy of the gene, you still have your good wild type version of the RB that can still work, right? That's why those early experiments with researchers fusing a cancer cell and a normal cell, they would see that the cancer phenotype would be neutralized. Why? Because in those particular cases, those cancers came about from loss of things like tumor suppressor proteins, right? So being able to restore the presence of normal tumor suppressor proteins like RB in those cases was enough to cause the tumor phenotype to revert, right? So in a case where you have hits happening at something like RB, we can talk about this concept of loss of heterozygosity, or LOH, which is a very important concept in cancer progression, right? Because hopefully what you realize is it's not just one hit on one copy of the gene, right? You need two hits, right? One hit would just create a heterozygous situation where you have one bad copy, but one good one, right? And you're heterozygous plus minus for the status of that particular tumor suppressor gene, right? The real trouble happens is when you lose the heterozygosity and you become homozygous for minus minus, right? That's why this term LOH, at first it seems kind of confusing, right? But until you realize what you're talking about is being heterozygous isn't necessarily enough to cause cancer, right? It's the loss of that heterozygous status to become homozygous for minus minus that opens the door for cancer to happen. So a harmful mutation or a harmful hit in one copy will make your cell, that particular cell, heterozygous, but it might still be okay. And keep in mind, because these are random events, we're not saying that at one given moment, every cell in your body, right, gets a hit in that one spot. I mean, that's kind of silly. What we're talking about is one particular cell gets, due to bad luck, a particular hit right at that spot in its genome, right? But that single cell might still be okay because it has the other copy. But in sporadic cases, right, that first hit might be something just like as a point mutation, and that second hit might be something like a random translocation event, right? Because it's probably very unlikely that it'd be another point mutation, right? And if then that cell starts dividing and that cell passes the bad copies of the gene in such a way that they both get passed to the same daughter cell, right, through mitotic recombination, then that's one way that loss of heterozygosity can happen. So let's look at this here. You have one copy of the chromosome from mom and one from dad, and dad's has the mutant RB allele, right? So during S phase of this cell, so this cell is heterozygous, so it's still okay, but during S phase, the chromosomes duplicate, right? And it's supposed to be what? Like the left copy of the red one and the left copy of the green one go to one daughter cell, and then the right copies go to the other cell. And therefore, the heterozygosity is still preserved in the daughter cells. Right? But you can have these weird sort of mitotic recombination events that happen such that the arms cross over. I mean, they're not supposed to, but hey, if you have chromosomes close together, it's always possible that the arms can cross over. Right? And in that sense, you might either retain the heterozygosity in the daughter cells, or the chromosomes may segregate in a way that now one of the daughter cells, right, is now lost its heterozygosity. It's become minus minus. And this daughter cell then might be the start of some transformational event, right? This daughter cell might be the single starting cell that's bad, that's going rogue, that ultimately might form a tumor that leads to cancer. One way you can study this is by tagging or labeling the DNA with certain types of markers. So researchers often, when they want to try to follow or understand what genes are important for loss of heterozygosity being the reason for cancer, right? They will have certain sort of markers around the genome that are kind of just randomly chosen, because after all, each chromosome is just a big long piece of DNA, right? You don't even know necessarily what genes are on there or if any of them are even relevant. But let's say you had chromosome 13, right? And let's say the retinoblastoma, the RB gene is over here on the left, this region of it. And then you just have a random marker, this AAGTC that is here in dad's copy, right? In mom's copy, maybe it's a polymorphic site. So this is AAGCC, right? So that way you can tell, you can follow what's mom's copy, what's dad's copy, right? By following that, you can identify if both of these markers end up appearing in one of the daughter cells, right, or in the tumor, for example, you can say, oh, this region of DNA somehow got segregated improperly, right? And now we're finding this improperly segregated chromosome in these tumor cells. So therefore, right, other genes in this region might actually be responsible for the fact that these are tumor cells that we're looking at. And that's how you can trace back and say, oh, yeah, the RB gene is here, right? So potentially then the defective version of the RB, right, that was traveling pretty close to this marker here, right, because you now see that that marker is retained in the daughter cell, and the daughter cell is now homozygous for this marker, you can imply that it's also homozygous for other genes that travel within that physical fragment of DNA. Because remember, our genes are not, you guys know this, our genes are not passed on as individual discrete genes, right? Our chunks of our chromosomes are passed on through meiotic recombination and things like that. So any gene that travels and gets passed on, it's very likely that the other genes that share that same sort of location on a chromosome also get passed on. One way you can also study this is by looking at restriction fragment length polymorphisms. So again, it's that same idea of having certain polymorphic sites that can be used to identify different regions of chromosomes that are being passed on that may contain then the genes that you want to focus on as candidates for explaining how this tumor formed. Here we have, for example, the maternal chromosome. Let's say mom's version of this region of the chromosome is a GAA-TTC, whereas dad's version, right, is GAG-TTC, right? So again, these are polymorphisms, right? Variations that just happen, and it can be used to trace your ancestry and your lineage. So in this case, maybe this polymorphism happens to be, because it's a GAA-TTC site, it's a site that you can cleave, so you can extract the DNA, genomic DNA, and then in a test tube, do a restriction enzyme digest using some restriction enzyme like EcoR1, right? Because EcoR1, this is the site in DNA that EcoR1 cuts. So therefore, if you have mom's version of the allele, right, the EcoR1, let's say, will cut here at the end and here at the end, because over here, let's pretend there's also EcoR1 sites on either end. But importantly, it'll also cut somewhat in the middle here, where you have this EcoR1 site as well, right? If you have dad's chromosomes mixed in there, right, he doesn't have that middle EcoR1 site. So as a result, he'll only get cuts at the end here and cut there. And now if you go ahead and you use a probe that recognizes a particular random region of this chromosome, right, and you use that probe to identify the sizes of the fragments that are being run out on a gel, for example, you can tell the difference between the two alleles, right? Because the allele that just has one big fragment here, right, that's dad's allele, right? That's then labeled by the probe. But in mom's case, you would just see a smaller fragment, the right side over here, labeled with the probe. Okay, and so that's one way you can run these big gels and actually identify, right, if in certain tumor samples you have evidence of loss of heterozygosity, where you only have one parental allele or the other, rather than a mix of the two. Okay. So different gel patterns in normal versus cancer cells can implicate loss of heterozygosity has happened at a particular genomic site in tumor cells compared to the normal cells that originally gave rise to them. So loss of heterozygosity is a very useful tool for trying to identify different regions on different chromosomes in our genome that are associated with different types of disease. So for example, this data here, this is looking at colon tumors, right? And let's say that you are an oncologist and you're interested in identifying, hey, what genes throughout the human genome are somehow important in the development of colon tumors, right? So you can do a big study where you look at lots and lots of colon cancer tumors, and you look for evidence of loss of heterozygosity, right? In the form of, let's say, deletions, right? And so that's what this map is. So across all 22 of the chromosomes here that are included in this, they looked at evidence of allelic deletions or loss of heterozygosity happening in the Q arm or the P arm of these chromosomes, right? And you can see by collecting this data, you can see that certain chromosomes like chromosome 8, 17, maybe 18, there is very compelling evidence that loss of heterozygosity involving those chromosomes is associated with having colon tumors, right? Because again, remember, this is starting from colon tumors, looking at them and looking for this evidence, comparing them to normal tissues, right? So that might help you narrow down your search for colon tumor specific genes to chromosome 8, 17, or 18, right? It's a good way to start building a hypothesis and start looking for things. This is the last slide for today, and this is a more modern approach for looking at loss of heterozygosity, right? So now these days, because of genome-wide sequencing, you can sequence the entire genome of a cell, right, by basically canvassing the entire genome with little probes that should sit down at specific spots along all the chromosomes, right? And if you've designed these probes well, I mean, you don't have to design them, they're available now, right, in these libraries that you can order. You can identify in certain samples of genomic DNA that you've extracted from tumors, let's say, are there regions of certain chromosomes that are missing, right? And because you're using tiny little probes now, right, you can be very specific. So here is an example of deletion mapping to identify potential tumor suppressor genes. I don't remember what cancer this is looking at, but in this case, they've narrowed their search down to a region of chromosome 9, 9p21. Right? So as you go across from left to right, each one of these little dots is an individual probe. Right? I think the way that it's color coded is actually the reverse of what you might intuitively think. In other words, white in this case means no problem. There was good representation of probes binding to their target spot. The darker the spot is means, there were fewer examples of the probes binding there, right? So the darkest ones, blue, means, oh, in these particular regions, the probes didn't bind. That means there's DNA that's missing, right? So each one of these, if you go from left to right, is a region of this chromosome, okay? And as you go down, right, each row itself, let me rephrase that, right? The columns going down, each one of these dots is a different probe, right? The rows going across, each one represents a different tumor cell line that you're looking at. Right? And remember how we looked at these kind of neat heat maps before that are only really doable now with the assistance of software and computers, right? This is a good example of that. So if you've done this sequencing, what you can do is you can line up all these different tumor cell lines and look at this 9p21 region. And what you can clearly see is that there's a region of 9p21 that's missing in a lot of these tumor cell lines, right? And the software has arranged it from sort of broad areas missing down to very narrow areas missing. Because remember, these are different tumor cells. They would have different types of deletions in them. Some of them have big chunks of 9p21 missing, right? But if you line it up from broad to narrow, it really helps guide you down to a particular spot. Right? And this particular spot happens to be in this CDK gene. Right? This gene is a gene that encodes for one of those cyclin-dependent kinases, that contribute to the normal functioning of the retinoblastoma protein, right? Okay, so that just gives you some sort of introduction to how we can look for tumor suppressor genes and also the importance of that. So next time we will continue looking at other examples of tumor suppressors and the different ways that tumor suppressor proteins work, okay, to actually control potential oncogenic pathways. All right, so let me go ahead and stop this.